Heath Turnquist. I'm from the University of Pittsburgh, and really what helped my career and my research at this meeting is really an opportunity to interact with people who are, doing immunology, but it's outside my field. So we have transplant. I do transplant immunology, and so there's a little bit of that here at AI, but it's really cool to see the people in infectious disease, what they're doing. Metabolics, just basic T-cell immunology, and just then be able to apply that in my field when I go back to Pittsburgh. What I love most about what I do is, for one, it feels like immunology is involved in everything, and so it's, I think I've really picked the right field in that, you turn on the news, everything is touched by immunology, and just it's kind of like playing with Legos for big people. We get to use mice that have knocked out proteins. We have mice that have cells that glow red, and then how they interact with cells that glow green. It's a lot of fun. It's cool to learn the knowledge, but at the same time, it's just cool to see it happen as well. My life before I started working in my field, so I was getting an undergraduate in psychology, and then with the idea of going to be a clinical psychologist. So I really like to study how things interact, and how that drives outcomes. I started doing the clinical rotations, and it just kind of wasn't my cup of tea. At the same time, I was working at a veterinary diagnostic lab, which I was looking for parasites in cow brains and sheep brains, or looking at parasitic worms. But I really liked the lab work, and so I stayed on, got a biology degree, and then went on to get a PhD. And so it was, that's what drove me to where I'm at. And I did a PhD in tumor immunology, where we were doing dendritic cell vaccines, trying to get the immune response to respond to a cancer, which worked really good for about 40 days. And you could see the tumor start to grow again. And so kind of as I was finishing up my PhD, I started looking into what cells were causing that. And it's these regulatory T cells, these immature regulatory myeloid cells, which really got me interested in those. That's how I ended up in transplantation, because they were the ones that were trying to apply that knowledge, the regulatory responses, to help transplant people. And so the idea was to do that for a few years and then go back to cancer. But I really liked working in the models and just stayed in transplant. The aha moment was really just seeing, you're excited because you're going to cure cancer or whatever with these vaccines, and just to see it work. And then, these tumors start shrinking, and then it started to grow again. And the aha moment was really all right, what cells are doing this? And it was these FOXP3 regulatory T cells, immature dendritic cells. So this is 10, 15 years ago, where that was just kind of starting to be new knowledge. And it's aha, these guys are really important. These are powerful cells. They're detrimental in cancer, but can you apply them to autoimmunity or transplantation? And so that was kind of the aha moment. It was I really want to learn more about these guys. The testing that we've done in the field really to advance immunology is, most of our work has really relied on knockout mice. So we'll knock out the cytokine. The cytokine we work with a lot is interleukin-33. It's kind of unique that it's actually not secreted by immune cells, but it's made in the barrier tissue, so when that's wrecked or broken, it's released. So it's kind of a signal for damage. And so what we've really done is looked at, all right, how does it tell the immune system what to do? And so what we're really finding is actually it's kind of like that first signal, but then it'll help shape what's going on around it. So if you have a virus, it'll help the antiviral response. If there's no pathogens there, then it'll help tissue repair. So that's one way I think we've really kind of changed how people thought about interleukin-33. At first it was this Th2 driver, so it was good to get rid of parasites. But our work and the work of others have really showed that it's good to actually fight off a virus. It's good to fight off cancer, and if you don't need it to do that, it seems to be really good at fixing tissue, doing tissue repair. What motivates me in my field really is just kind of the quest for new knowledge, or the curiosity. I think it's just cool to make new discoveries. That's a lot of it. And then actually the students too. I really enjoy training graduate students, just to see them come in, really not have an idea how to do an experiment, not how, technically not have all the pieces, but just see them, over the years come together to where the point where they're self-sufficient and they're bringing you in data and they're excited about it. So really to, spread knowledge that way gets me excited and makes me come to work.